Super. Sir, Kiran sir, very good morning, sir.
Good morning, sir. Good morning, sir.
Sir, first of all, very thanks and gratitude for accepting wholeheartedly to conduct this international virtual learning series, sir. Thanks
Thank
for
you.
your kind acceptance, sir.
Thank you, sir.
Thank you, sir. அன்பு சார் ஸ்டார்ட் பண்ணலாமா? வணக்கம், அனைவருக்கும் இனிய காலை வணக்கம். First of all, I put forth my sincere thanks to our management for accepting and helping hands wholeheartedly to conduct this international virtual learning series. It is honor and a privilege for me to welcome you all on the behalf of our AKCP for this 28th series. Today we are very proud to have an eminent personality. அன்பு சார் கேக்குதா? We are very proud to have an eminent personality, well-known academician, and honorable resource person, Dr. Professor Dr. KIRAN CHANABASAPPA NILUGAL, Faculty of Pharmacy, Management and Science, University, Malaysia, for his kind consent to share his knowledge, experience, expertise to deliver the lecture on DMPK studies and overview. It is great pleasure for all of us that he could find time to be with us in spite of his busy schedule. I take this opportunity to convey my sincere thanks to you and your esteemed management for accepting our request despite of your busy schedule and for being a valuable part of this session. Most welcome, Professor Dr. Kiran sir. Kiran sir.
Thank you, sir. Hello.
Thank you, sir. Next, I welcome all the professors, faculty members, and my dear enthusiastic students for today's session. My dear students, I hope this session will be a good platform for our students to explore the knowledge about the drug metabolism and pharmacokinetic is a scientific discipline once primarily associated with the safety evaluation in drug development that has last 2 decades become a core discipline within the drug discovery. So students kindly utilize this great opportunity. This is the first and foremost needed for your career. All the very best, my dear students. Once again, I welcome you all. Thank you. Welcome, sir. Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir. Please, you can proceed, sir.
Okay. Thank you, sir. Can you hear my voice, sir?
Yes, sir. Clear, sir.
Okay, sir. Thank you, sir Venkatesan, sir. Thank you for the invitation. Okay, it's a great honor to give a lecture on my topic is DMPK studies and overview. If my voice is not clear, let me know. So I can check. So I'm working. My voice clear, right, sir? Hello.
Sir, yes, sir. Clear, sir.
Okay, sir. So
Here, sir. Here.
yeah. Okay, sir. I start my lecture.
Sure, sir.
So I am Mr. KIRAN NILUGAL. So I am Master of Pharmacology, and I submitted my thesis, waiting for my doctorate. So I have an industrial experience in Zydus Research Center. I work in Vimta Labs, and now I am working as a lecturer for more than around 6 years. I am working as a lecturer in School of Pharmacy, Management and Science University. So my lecture today is based on my experience in DMPK. So I work in drug metabolism and pharmacokinetics. I want to share an overview lecture on DMPK. So how drug metabolism and pharmacokinetics has a role in drug discovery. So, so, uh, we, I continue my slides. So, as you know, the journey of NSNC is new chemical entity. So, when the molecule is synthesized in the lab, so we need the pharmacological, whether the drug is having pharmacological effect, whether the drug is having any pharmacological effect based on the therapeutic category. You have the cell biology, and we have a role of DMPK where you check the drug metabolism and pharmacokinetics. So, because this department, everybody know what is pharmacokinetics, where you study the absorption, distribution, metabolism, and elimination. So here, most of the drug discovery, okay, they call the department in drug discovery, they call as DMPK, Drug Metabolism and Pharmacokinetics Department. So where the scientists, they work on the pharmacokinetic studies in animals, pharmacokinetic study in vitro, and they also work on the analytical instruments like HPLC and mass spectroscopy. So this is about the DMPK. So you can have, we also have the toxicology studies. We check the toxicology samples. We do the method development for the HPLC and mass spectroscopy. So later you have to file the IND investigation new drug filing, and you have to file the file, and then later you have to develop a formulation and you have to do the clinical trials. So as we know that preclinical trials we usually do in vitro and in the animals. So preclinical trials, after you get permission for approved for the preclinical studies, are feasible for the clinical trials. So so we discuss how, as a DMPK department, how it is having a role in the drug discovery mechanism or the drug discovery process. So you can see the role of DMPK from this slide, where you already know the target identification and validation. So what is your target for your drug discovery? So you have a lead discovery. What is, because we have so many molecules. So in that you select one. Okay, this is my lead, and then later you optimize your lead. Then you select your candidate of drug, and then you go for the preclinical development. So this target identification and verification, okay, so depends on your dose, depends on your study. So when the molecule, you already know that this molecule is having the pharmacological effect. So the drug main thing is you have to check the pharmacokinetic profile. So most of you know about the pharmacokinetics. So where your drug should be absorbed, okay, then only there is a pharmacological effect. If your drug is not absorbed, when you take the drug orally, it is not absorbed in the stomach, it doesn't reaches your blood circulation, so there is no chance of pharmacological effect. So that's the role of this, our drug metabolism and pharmacokinetics. So here we have to study different PK properties. You have to screen many drugs. We have many drugs, in that we have to select the best PK profile drug, and then later you can go for further studies. We also need support from the medicinal chemistry, pharmacology, and because to know the structure of the drug, we need to know the pharmacology of the drug, then we see the pharmacokinetics. So this we then from that we predict our pharmacokinetics. That is helpful, okay, promoting your pharmacokinetics and pharmacodynamic actions. So as you can see here, so our DMPK activities is very important in drug discovery during the preclinical development. If your drug doesn't have any good pharmacokinetic profile, so your drug cannot be go further preclinical studies. So, so your drug can be rejected during these studies. So because it doesn't have the pharmacokinetic properties. Okay, so when you study the, when you work in the DMPK department, it's one of the department in the drug discovery. So this DMPK department, you can find most of the contract CRO, contract research organization, those who get the studies from the other companies, they have this DMPK department. So when you give the molecules to them, they will do all the testing and submit the data to you. So it is in most of the research center and most of the preclinical CRO where they do these DMPK studies. So when you come to the DMPK department, so we have in vivo studies. In vitro is inside, okay, the lab. In vivo is inside our body, that is using animals. In vitro, we prefer in vitro studies because it is easy to do and you can easily predict the pharmacokinetic parameters. So rather than going to in vivo where we require animals are expensive and maintenance of the studies. So when you do your in vitro, okay, your drug is having good good absorption, good solubility, good plasma protein binding, good metabolism. Your drug is stable in the plasma, in your blood, and your drug is involved with the cytochrome enzyme. These are in the liver. We have the liver cytochrome enzyme where we can get these enzymes we get from the animals and the human. So this you can put the drug inside this CYP enzyme we call a cytochrome pooled enzyme and test whether the enzyme is this. We have different type of isoenzyme whether your enzyme is going to inhibit the drug or induce the drug. So this is very helpful for the drug-drug interaction. So in vivo, so if okay in vitro your drug is very good, so we can do the in vivo studies. We do the ADME, we do the absorption, we do the distribution, metabolism, and elimination. So all these parameters we do in the animal. Hello, my voice clear, sir? Ambu, sir? Hello, hello, sir. Okay, okay, sir. So we have the PK parameters. You know the pharmacokinetic parameters. You already have been studied pharmacology and pharmacokinetics. We have hello, sir. Hello. Okay. My screen okay, sir, because I already uh, can see, sir, my screen okay or not? Okay, sir. So you study the PK in vivo study ADME. We have the PK parameters like Cmax, Tmax. You have been studied and toxicokinetics where we study the toxic dose. For example, you have to do a toxicity studies. So whether your drug is toxic or not. So we select the toxic dose and we do the toxicology studies in the animals. When we take out the blood sample and then we plot a graph, that is called as toxicokinetics. So TK study is also important to evaluate the adverse effect of the drug. So, so this is what we do in the DMPK. And when it comes to in vitro, so in vitro pharmacokinetics, so it's routinely used to predict the ADME, or we call as ADME, absorption, distribution, metabolism, and elimination. So in vitro assays are very faster and less expensive compared to the animal. So we have many in vitro assays that allow the cross species comparison between the mice to man. That means we have the liver enzyme. We can get this liver enzyme in the mice, rat, and human. So this we get from the human after the death of the human. We take out the liver and centrifuge and then extract these enzymes, the CYP enzyme, then store. The same way in the rat and mice. So we already have the enzyme, so we can do the metabolism studies in vitro. So no need to give the drug to the animals and check what is happening in the metabolism. So metabolism we can do in vitro. So these are very helpful. In vitro pharmacokinetics can determine your efficacy or your pharmacokinetic profile of your drug. So this is about the in vitro studies. So when you work in the DMPK department where you deal with your in vitro studies, so you have to do all these studies. You have to learn these studies like the solubility of the drug, the stability. Because when you take the drug orally, your drug should be stable in the gastrointestinal fluid. You should also check whether the drug goes inside the blood, then whether the drug having plasma protein binding is very important. We can do by different methods, in vitro methods, like rapid equivalent device. We do ultra centrifugation for the plasma protein binding. We can do the metabolic stability about the drug metabolism. You study phase 1 metabolism, phase 2 metabolism, that you can study by using the pore liver microsome. So this microsome is available, and these are from the mice, rat, dog, monkey, and human. So these are already available. So you just take and add your drug, incubate, and then find out what is happening to the drug. So plasma, we have to do whether your drug is stable in plasma or in the blood. And also you can do the inhibition, whether your drug is inhibited by cytochrome enzyme, maybe 3A4, 2D6. You have to identify the iso enzyme. So we can identify by the induction and inhibition studies. So these are the list of studies we do in vitro pharmacokinetics. So I will be discussing some of the studies, okay, so that is done in vitro. So in vitro, we do, the first one is we do this use of polarized CACO2 cell line. So these are the cell line. So we know that when you take the drug orally, the drug is absorbed in the intestine. The major route of absorption of our drug is intestine. So this CACO2 cell line is human adenocarcinoma cell of the colon. Okay, and that differentiates into monolayers and as a functions of small intestinal villus epithelium. So these cells have permeable filters that can predict. So rather than giving the drug to the animals and check whether absorbed or not, so we can use the cell line and then we can see whether the drug is absorbed. So you can see from this is cell line, and in the next slide you can see here, this is what we follow here. We have a monolayer layer where we have, this is your cell line. We add the drug solution here, and then after absorption, the drug should go here, B. So, so this is your cell monolayer, this is A and B. So after your your drug is absorbed, it comes to B. So based on the concentration in the A, what is the initial concentration, and later after absorption, what is your concentration? From that, you can predict whether your drug will be absorbed in the intestine. So rather than you give to the animals, you can check here. So this will save your time and save the cost. So this is one of the in vitro testing that we use the CACO2 cell line to study the permeation, that means the absorption of drug. So next is we have everybody know what is plasma. So you can see the whole blood. So we take out the whole blood and then centrifuge, you get the plasma. And this plasma, everybody, so we know that plasma contains the different plasma proteins like albumin, globulin, fibrinogen, and hydrolytic enzyme. So we also have enzymes like esterases, lipases, and other enzymes. So so to check the plasma protein binding. So when you take the drug, we know that most of our drugs are bound to the plasma proteins, and this binding to the plasma protein is a reversible process. So the drug is released slowly for a period of time, and that determines your half-life of your drug. Okay, when it is more protein bound, okay, so then you have more drug remaining in the body for a longer time. So this is the plasma and the whole blood. You can see how you centrifuge, you get the plasma, and this is the plasma protein binding. So this is very important because we cannot find the plasma protein binding. We just give the drug and check. So this in vitro method, we have the two plasma major proteins like human serum albumin, and we have alpha 1 acid glycoprotein. So simple, we say glycoprotein, alpha glycoprotein, and human serum albumin. So these two plasma protein, these are the plasma proteins. Your drug is going to bind to this. So when it bind to the plasma protein, so how to check in vitro? So you can check in vitro by the in vitro methods in the pharmacokinetics in vitro. So In the next slide, you have these methods. Okay, that is we call as rapid red RED is rapid equilibrium dialysis, and another method is we have ultra filtration. So this equilibrium, where you see from this slide, you have the plasma and the drug. If we get the plasma, we collect from the animals or the human. We get the plasma, so you add the drug, and you see what happens. This is the other layer is a buffer. We add the buffer, and then later you find how much drug is bound and how much is released. Because when it's bound, okay, releasing comes to the buffer layer. So you have the C total and the C free. So this is one method. Another is ultrafiltration, where you filter here. So the free drug comes out here. So from this, we can find the percentage protein binding, okay, by the concentration, the total and the free. We calculate and we find, okay, this drug is 90% plasma protein binding. This drug is 85%. This drug is 60%. So depends on your chemical or chemical entity or the new drug you synthesize in the lab. So based on that, in vitro, by doing this, you can know whether your drug is bound to the plasma protein. So this is how is some one of the in vitro testing for the pharmacokinetic profiling. So, so this plasma protein binding is very important, because you see that most of the drug, you read your SINs, you read many drugs, you can see the protein binding, okay, is 95%. You read the pharmacology of drug, maybe any drug, you see the plasma protein is 95% protein bond. So the free drugs available is protein bound drugs serve as a depot. So longer half-life, and the drug is released slowly for a longer duration of action. So, but still there is a debate whether the protein binding should be considered. But most of the drugs are highly protein bound. So they have more drug in the body, and it is released for a longer duration of time. So this is the plasma protein bonding. You can find the plasma protein binding here, the free drug, and when it is bound to the plasma proteins. So you can find the difference. So here, when the drug is bound to the plasma protein like albumin, so you have more drug. Okay, that means the drug remains for a longer time inside your body. So there is more action of the drug. So this is about in vitro plasma uh, protein binding. We study in vitro, and now you can see from this picture or from this diagram, so you have your molecule, you have sufficient in vitro. That means, okay, you have good absorption, plasma protein, enzyme inhibition, enzyme induction, metabolism. Okay, your drug is okay. Now we can proceed to in vivo. In vitro, okay. If in vitro there is no absorption, no protein binding, so we don't go for the animal study. So in vitro, okay, then we pass the drug to in vivo, you do in the animals. So that is in vivo. So after you confirm with the in vitro studies, okay, this drug has a potential, then you go for the in vivo study. So you can do, we already confirmed by in vitro metabolism, in vitro permeation, drug-drug interaction by using the pooled liver enzymes. So then we proceed to the in vivo pharmacokinetics that we do in the animals. And this, most of you already studied about the plasma level time curve. This is what we plot the graph, the pharmacokinetic DMPK. So every drug, when you administer the drug, then we collect the blood sample for different time intervals, and then you get a graph, that is oral absorption graph. So you know what is your drug is absorbed, okay, distributed, metabolized, and eliminated. So this is a common oral drug, typical pharmacokinetic profile of oral drug. So you know what is Cmax, time to reach the maximum concentration inside the blood. We have the maximum concentration Cmax, and Tmax is time to reach the maximum concentration. So you know this is a typical plasma drug concentration curve. So this we give orally because oral most of the drugs are taken. Oral route is most widely preferred route. So we have this oral profiling of the drug. So uh, now we already discussed until now we discussed about in vitro. So your drug is good drug candidate for in vivo. So we can go for the in vivo studies. So for the in vivo studies, we are using blood samples. Why we use blood sample? So because it is a central compartment. Because when you take the drug goes to intestine, absorb, goes to the circulation, that is your blood, and it has a definable relationship to tissue concentration. And because we know that when the drug goes inside the blood, your drug will go wherever there is more blood circulation. So your drug will be more in the liver, more in the heart, more in the lungs, where there is more blood. And this is easy to sample. We can collect the blood easily. We can also analyze. We can see how much drug is there in the blood. So this is why we take the blood sample in the pharmacokinetic studies in the animals as well as in the human. So this is we prefer these blood samples. So now you can see from this slide where because we are doing the preclinical DMPK preclinical studies, so we use animals. So we can use these animals. We can use mouse, rat, dog, and monkeys. So, based on the FDA guidelines, we can use one rodent. Rodent means rat and mice. Non-rodent, you can use either rabbit or dog. But the FDA, they ask dog. Okay, they don't prefer rabbit. So, some of the studies, they prefer in the dog as a non-rodent. And some of them, they prefer as a rabbit. But most of the studies, they do in the rat and the rodent. Or you can go for the non-rodent is dog. So you should have the pharmacokinetic data for the rat and the dog. Then only your drug can be approved. Otherwise, it is not approved. So this is the animals we commonly use for the pharmacokinetic studies. So now, when you study the pharmacokinetic study, so we everybody know we do in the in vivo, we do in the animals. So we can do different type of studies. So we maybe you know that pharmacokinetics we just give the drug and then take the blood sample. But we can do many studies. We give a single dose to the rat and then check. Take 0 minutes, then 10 minutes, 20 minutes until 24 hours. Collect the sample, then you plot the graph. Analyze the sample, then you get the concentration, then you get the graph. You can do the bioavailability, amount of drug reaches the systemic circulation. You can also, we do all these studies like how food is affecting the absorption of drug. So we give the food and then the drug, fed rats and fasting rats. So we do the two groups. So what is the difference? And also we do the gender, that is we do in the male and female rats or mice. We can do the elimination studies. We can do species, that means the rat, mice, and dog. We can do excretion. That means in this pharmacokinetic studies, we collect this urine and feces from the rat, urine collection and feces collection from the rat, and then we process the sample and then we check what is the concentration of drug in the urine as well as feces. So this is so that you know what is how the drug is eliminated through urine and feces. These are the pharmacokinetic studies we do in the animals. So we need all these studies to support whether your drug can be approved or not. So now we have this toxicokinetics because your drug also you should check the toxic dose. When you determine your toxic dose, then you take three doses, that is low, mid, and high, and you do a simple toxic studies. You can do 28 days, and then you collect the blood samples. Usually, the pharmacology and toxicology department conduct these studies, and they send the sample like day one blood sample. We go and collect the sample. So when we collect the sample, so day 1, day 14, day 28. So when you collect the sample, we analyze and get the graph. So how we see how toxic dose, what is the toxic dose, how much drug is there in the blood. So this is how we collect we have the toxicology department, and that study we do is called as toxicokinetics. And we can they do the acute toxicity and the chronic toxicity they can do for 1 year. They can do for 3 months, 1 year, depends on the toxicological studies. So you can do the tissue distribution studies. That means you can give the drug to the rat and then later sacrifice after it reaches the Cmax, sacrifice the rat and then take out the different organs of the rat, brain, everything. You take out the rat brain, you take the liver, and then make a homogenate. Homogenate is like a juice of the organ. Then later you analyze, extract the sample and then analyze in the HPLC or mass spectroscopy, and then you get your concentration. So we find, okay, this liver has this much concentration of drug. So this determines the toxicity of your drug. We also, they do the histopathology department, they do study the histology. They take the liver sample, they cut the liver sample and then process and then make the slides and check what is happening to the liver cells, the hepatocytes, what happens to hepatocytes, what your drug is doing to whether the drug is having liver toxicity or not. So this can determine by doing the histopathological studies. So next is this is what we do in the pharmacokinetics in vivo. But now in vivo means we have to collect blood. Okay, so after you collect the blood, we have to process samples, send to the HPLC, the bioanalytical, where you collect the whole blood. We collect the blood, then we centrifuge, then take the plasma, then you have to extract the drug from the plasma. So why plasma? Because in the whole blood we cannot extract the drug. Difficult to get the drug, extract the drug from the whole blood. So we take the plasma. So how to extract the drug from plasma? So we add some solvents. So you can add methanol, simple precipitation reaction. You can add methanol. You can add acetonitrile, that is methyl cyanide. You add, and then there is denaturation of the protein. Then you centrifuge, you take the supernatant layer, and then you inject the samples in the HPLC or mass spectroscopy, that is liquid chromatography with mass spectroscopy. So this is how we analyze. From we take the blood sample, then extract, then find the concentration, then we get the graph in the HPLC. From that, we calculate the concentration, then we plot the graph. We saw the graph before, we get the pharmacokinetic graph, the Cmax, Tmax, everything after we do the analysis. So this is how we do the blood sampling. And the next is measuring the drug levels in the tissues. We can also measure in the tissues. We can take the brain sample. This is done in the animals, not in the human, rat, mice. So we take the brain sample and then we can centrifuge, and then we can see the concentration based on the weight. We weigh the brain tissue, what is the weight, and then from that, okay, we centrifuge, and then we get the concentration of the drug in this. This is the weight and this is the concentration. So this is how we can also measure the drugs level in the tissues. We can measure in the liver, in the brain, in the heart, in the lungs. Okay, so this is how you measure the drug levels in the tissues. So okay, next is we go for the analytical techniques, because when you work in the DMPK, DMPK is involving the pharmacologist and as well as the chemistry. Pharmacologist and chemistry is involved, both involved working in the DMPK department. So pharmacologist does the in vitro studies, in vivo studies, and also help for the analytical techniques. The chemist develops the method development for this drug. Okay, what is the method? Okay, what is the mobile phase? What is the stationary phase we use? What columns we are going to use? All this we are going to use is done by the chemist, and also sometimes we have to work both. We have to collect the blood sample, we have to run the HPLC, Okay, so when I was working, we used to run HPLC, also collect the blood samples, everything. We, you do all, one person doing all the work. So you have exposure to collection of blood, also sampling, also you can handle the HPLC and LCMS. So now, when you take the blood sample, so how to extract the drug? Because we saw in the previous slide, you just put a solvent, that is plasma protein precipitation. So you get your drug easily. If you cannot get, after you extract, you put in the, you run in the HPLC, you don't get the graph. You cannot get the peak. So there is no drug because your drug is not able to extract from the blood. So you can go for liquid extraction. You can go for the solid phase extraction. So these are the extraction methods. So the first one is the plasma protein precipitation. So this is simple method. You take the blood, centrifuge, then add the solvent. Then you can have, so you can take the superman layer and then you can inject in the HPLC. So here depends on the gradient, depends on the column. Okay, so we can use different columns, C18 and C9 columns. So mostly the chemist will do all this. They analyze which column, the mobile phase, everything they analyze, and then they they can get the concentration of the drug. So this is extraction method. So extraction method, how we extract the drug from the plasma. So we don't extract the drug from the blood. We cannot get the concentration of blood by extracting. We cannot extract from the blood. So we take the plasma. So that is how you, the extraction method is your plasma protein precipitation. The next is liquid extraction. So depends on your drug whether it is acidic or basic. So you can use solvents like ethyl acetate. We can use hexane. This is like a trial and error. So we don't have a definite because your drug is a new drug to synthesize the lab. So we have to use different solvents. We should check with this column. We get the graph. So this technique is when you are not able to get the concentration or the peak, your graph or your peak from precipitation reaction. So you go for this method. You try this method. So maybe you can try this precipitation, liquid, another solid phase extraction. Then you check, okay, how what is the difference of drug we extract from these different methods. So this takes this liquid extraction takes your time. Sample preparation is very long process, and then we have to add different solvents. Then centrifuge. Then okay, so dry it, and then later inject in the HPLC. So this takes a laborious process. So usually in the lab we follow the precipitation reaction because very easy to extract the drug. The next is you can go for the solid phase extraction. So solid phase extraction is usually for the bioanalytical studies, BAB studies, where they do in the clinical trials. So when they collect the blood from the humans, they go for this solid phase extraction. And this extraction is very good method and easy sample because we have a solid phase extraction. We have ready-made extraction tubes where we put the sample automatically extracted, and we can directly inject in the HPLC or in the mask. But this method, the whatever the filters we use is expensive. So when you want to run sample, you have to think about the cost, whether we need solid phase, whether you need liquid phase, whether you need the precipitation method. So these three methods used for the extraction of the drug from the plasma. So these are the methods. And okay, in other studies, we can go for the pharmacokinetics. We have to do the dose proportional. That means, for example, you give 100 mg of drug to the rat, so you get the graph. Then you give 200 mg, then you give. So there should be a dose proportional. So this, my, can you see the slide? This slide okay or not? Visible. 1 minute. Okay. I think. Can you see now my slide, sir? Okay, sir. Okay. So this is about. Okay, thank you, sir. Thank you. So this is the graph. Okay, where you can see the dose linear. So you give one dose, then next dose. There should be a linear curve. So if there is no linear, there is no dose proportionality studies. So this also we do in the animals. So so you can see the two graph. Okay, one that is you can see the drug dose we give different dose and check. So you have we get the increase. We check the AUC and we check the Cmax. So there should be a dose proportional. But if you see these two graphs, there is no linearity here. So your graph like this. So why there is not non-linear? Because maybe you have problem in the absorption, saturation of absorption, metabolism, and other factors that will cause this non-linear pharmacokinetics. So this studies also we do in the DMPK department where we test in the rats and mice. So we give the drug and check different doses. We check. So this is my end of the presentation. So I have already told from the previous from the slide number one. We discussed about DMPK. What is the role of drug discovery? I discussed some in vitro methods and some in vivo methods, and then about the extraction method. So this is what you when you work in the DMPK department. This is what you have to do and contribute to the drug discovery. So you're working like your drug metabolism pharmacokinetics profiling of the drug is very important in the drug discovery process because your drug should be absorbed, then only there is pharmacological action. Sometimes when we do the studies in the lab, sometimes we the drug whatever sensor in the lab, so there is no absorption. Then later we check, we sacrifice the animal and check the drug already is in the stomach. It is not going to the intestine. It is remaining in the stomach. So from that you can find out whether from these studies, in vitro and in vivo studies, we can determine whether your new drug is can be further do for the clinical trials, or we can go for the further studies so that we can have a good drug for the clinical trials. So that is about my presentation on drug metabolism and pharmacokinetics, and thank you very much for giving me the opportunity to present. Ambu sir, hello. Yeah, so this is my slide. Already I finished my presentation, sir. Any questions? Please ask. You can ask. Yes. Pharmacokinetics, the success rate is not much, sir, because we in the last time I worked in the Zydus Research Center, we get around 10 molecules every day to do the pharmacokinetics. Maybe in that 10 molecules, maybe one only is having a good pharmacokinetic profile. But later, when we send, they do further pharmacological studies, maybe they may fail. So the success rate is lesser. Uh, how for the pharmacokinetic or PK studies, we can do in vitro, in vivo. We can do based on your drug. We can do maybe by 1 week or 10 days. We can have the time to do your drug, but that depends on your synthesized drug, whether your drug is good or not. So basically, if the drug is in vitro when you do the absorption and other studies, then we can suggest for the in vivo studies. Yes, sir. In vitro and in vivo, both studies, the DMPK department will do both the studies, sir. In vitro, we can also do the in vitro, in vivo correlations, sir. In vitro, in vivo correlation based on the data. Yes, sir. I can. Yes, sir. Yes, sir. Yes, yeah. But the possible. Okay, okay. I saw this. Some research person gene therapy is used for COVID. Okay. So still research is going on COVID, but we cannot test in the animals because COVID-19 is not in the animals. So we cannot test on the animals first. So we the drug therapy is to check to go for the in vitro method. In vitro to develop a new drug, we go for the in vitro molecular studies by using the virus. So we want to kill the virus, or we can kill the virus by using the drug. So if if it is successful, then you go for the human trials. But mostly now, when you see the COVID-19 drugs, all the drugs are already whatever we have. We have chloroquine, we have hydroxychloroquine, we have dexamethasone, we have all these antiviral drugs already developed, and there is no new drug. Uh, not developed. We are not having a new drug for this COVID-19, but there is more research going on to again repropose the drug. The drug, whatever available drug, is used for the therapy, and the research is going mostly to develop the vaccine compared to developing a therapy. So the animals we can select because we when you have a centralized animal house, so these animals are procured from the accredited laboratories, and we breed the animals in the animal house. So you can take. We have different models. We can use Vista rats. We can use Sprague Dolby rats. We can use mice. Okay, we can use Swiss albino mice. So either strains we can use. Based on the okay, sir. Um, based on the PK studies, we have to do in the small animals like rat and mice. So if the requirement, if we get a good profile by doing the study in the mice, okay, and then we see in the rat. So later you have to go for the higher animals, that is dog. That depends on your pharmacokinetic profile, sir. If you get a good pharmacokinetic profile, then you go for the higher animals, sir. Yes, sir. Poor metabolite. That depends on the again. We can improve by improving the formulation. We can change the formulation. We can improve that, sir. But mostly, if your drug is not having a good pharmacokinetic profile, so if it is not absorbed and it is not able to have the action, so we cannot. We can improve only the absorption. We can develop the new formulation so the absorption is increased. So there is more good PK profiling. So we can develop a formulation that can increase the absorption, sir. Okay, sir. Thank you, sir.
Thank you, Professor.
Hello, Ambu sir. Thank you very much, sir. Thank you.
Doctor, Doctor, Dr. Kiran sir,
Yes, sir. Thank you, sir.
Professor Kiran sir, we had a wonderful and informative presentation, sir.
Thank you very much, sir.
Sir, on behalf of our management, hereby express my wholehearted thanks to you and your management for giving us such a wonderful, informative presentation. Excellent presentation. And also, I want to extend my special thanks to Professor Dr. Pravina, management of Science University,
Yes, sir.
Malaysia, for wonderful arrangement of this session, sir. Thank you, Pravina, Dr. Pravina, madam.
Yes, sir.
Sir,
Thank you, sir. Sir,
Dr.
later
Kiran,
can you send some
Professor
pictures?
Kiran, sir,
Hello, sir.
another one more request, sir.
Yes, sir. Yes, sir. Yes, sir.
I hope this interaction will motivate the students and encourage them in active learning, sir, for DMPK studies, sir.
What
And
kind?
Let's have a good cooperation, sir, towards for the development of our students' career, sir.
Yes, sir. Yes. I cannot hear you properly, sir. Hello. Hello.
Yes, sir, can you hear?
Yeah, I can hear, sir, but disturbance, little bit disturbance.
Yes, sir. How?
Yeah, can hear, sir.
Yeah.
Sir, can you send the pictures
Sir,
of
in
the?
future also, let's have a good collaboration, sir.
Yeah,
That
sure, sir.
much.
Sure, sir. Sure, sir. Sure, we'll have collaboration, sir.
If possible, sir, kindly refer your friends, sir, for forthcoming webinars, sir,
Sure,
international
sir.
webinars.
So I'll refer, sir. I'll refer.
Sir, your friends are. Thank you,
Sure.
sir. Thank you so much.
Okay, sir. Thank you, sir. Thank you, Venkatesan, sir. Thank you, Ambu, sir.
Convey my regards to madam.
Okay, sir. I'll convey my regards, sir.
Yeah.
Sir, can you send the pictures you taken, the screenshot? Okay, sir. Okay. Thank you, sir.
Thank you. Take care, sir.
Thank you. Thank you, sir. Thank you. Thank you.
Thank you very much.
Thank you very much, sir. Thank you. Thank you.